Today we are going to talk about NGS. I assume that before getting into next generation sequencing, all of you should know about Sanger's sequencing. Sanger's sequencing. Sanger's sequencing was the technology that was used to generate the human assembly. And it took around 13 years to generate the assembly from 1990 to 2003. The take-home message from Sanger sequencing is that we need a primer for sequencing. NGS, however, these days can complete an assembly from a eukaryote in around 15 days. This makes NGS a high-throughput technology. Today in the class, we look at how NGS is a high-throughput technology. We are going to discuss NGS under the following heads. The first one is RNA sequencing, RNA-seq. The second one is variant calling. And the third one is genome assembly. The third one is genome assembly. Genome can be an RNA genome or a DNA genome. So NGS involves sequencing of both DNA and RNA. Today we're going to first talk about the chemistry of how the sequencing reads are generated in NGS. So let it be RNA or DNA or RNA. Initially, is fragmented into millions of pieces. This DNA or RNA is fragmented into millions of pieces. These pieces are now attached with an adapter. When I draw a fragment, it is a double-stranded molecule. These pieces are attached by adapters on both the sides. A1 and A2. When you have two adapters, A1 and A2 on either sides, in the process of attaching this A1 and A2 adapters to the fragments, there's every possibility that some of the fragments may have A1 adapters on either sides, and some of the fragments may have A2 adapters on either sides. Only those fragments that have A1 and A2 on either sides are selected. And this protocol of selection has been standardized. Now, as we know that primer is needed for sequencing, using the same fundamental, we now target both the A1 and A2 adapters to generate our sequencing reads. What does that mean? Now you have A1 here and you have A2 here, A1 here and A2 here. These are now targeted with primers. These primers, one in the forward direction and the other in the reverse direction. This, in the process of amplification, would generate sequences that is complementary to the bases that are present on the fragment here. Now, the length of this sequence that is actually synthesized and generated is now called as read length. Depending on the requirement, the read length now given by various companies is either 36 base pair or 50 base pair or 75 base pair or 150 base pair or can even be 250 base pair, or even 300 base pair. So what do we know now? By using a primer, forward primer, we can synthesize a fragment. In the process of synthesis, we can sequence that fragment. And the sequence length that is generated can vary from 36 base pairs, 50 base pairs to around 300 base pairs. And if the sequence is generated only from one side, means using the forward primer alone, then it is denominated as, let us say, if you generate 75 base pair fragment from one side alone, that is from this forward side alone, then this is referred to as 1 into 75. The moment you see something like this, you should understand that the reads for NGS are generated only from one side. And since they are generated from one side, they are called as single end reads. Single end reads. Now, you can also use your reverse primer to generate a fragment from the reverse direction. And if this again, you are sequencing in the process of synthesis, and if you are generating again a 75 base pair fragment, and now that you have generated fragments both from the forward as well as in the reverse direction, this is now referred to as 2 into 75 base pair. And this is now referred to as paired end reads. So now you clearly understand what are paired end reads and what are single end reads. If you are generating sequences only from one primer, that is the forward primer, it means that it is a single end read. And if you are generating reads from both the directions, from both the forward and the reverse primers that would attach to A1 and A2 adapters, then it is the paired end reads. And these sequences from the forward and reverse would correspond to the same fragment. Now, let us just assume what we are doing here. When you are targeting a particular fragment with a primer, let us say forward here, this primer F now has got a capability of attaching to all the adapters of different fragments. For example, this is one particular fragment I have shown you, right? Now you have some other fragment here. This fragment has also got what? It has also got, let us say, this is this particular fragment 2. I'm just denominating this fragment in red now. This is a fragment 2. Now it has got adapters on both the sides, A1 and A2, the same adapters, and you are targeting it with the F1 here and R1 here, or F and R, the forward and reverse. And now, the moment this forward primer binds here, this forward primer also binds to this particular adapter here. What I mean to say is, the forward primer that is targeted against one adapter A1 has got the capability of binding to all the positions of the A1 adapters across the millions of fragments. So, in the process of synthesis, millions of fragments are synthesized in one single go in NGS. Now, what is happening here? Since you generate reads from millions of fragments, there are a lot of sequencing reads that are generated across the entire genome that you have, and therefore you will be having a lot of data that is generated. And that is the reason why this is called as high throughput sequencing. High throughput sequencing. Now we'll look into, since you know what reads are now, we'll now look into how this fragmentation takes place. Fragmentation can now be dealt with. There are different methods of fragmentation. One, mechanical shearing. Mechanical shearing. 2, restriction enzyme digestion, RE digestion. 3, bead-based method. Bead-based method. Now, under mechanical shearing, you understand that there should be some pressure applied onto the DNA strands to be sheared into small pieces. The most common procedure is nebulization. And the gas used for nebulization is nitrogen. Why only nitrogen? This is because nitrogen is an inert gas. Yes, partially inert. You can ask me like why noble gases cannot be used here. Yes, 2 reasons. One, nitrogen is widely available. It depends on the availability. And as well, it is quite cheap. Whereas if you are going to go in for noble gases that are actually inert, right, the availability is quite meager and then they are very costly. So here, when you look at N2, it is often referred to as partially inert. Now, let us understand the electronic configuration of nitrogen. Nitrogen, if you look into, is atomic number 7, and its electronic configuration is 2,5. And now it can be written as 1s2, 2s2, and 2p3. And it has got orbitals here. If you look into these orbitals, 1s2 is completely full, 2s2, and if you're referring to p orbital here, right, p orbital is half filled. Just because the p orbital here in the second shell is half filled, this is partially inert. And that's the reason why you find N2 being used in the chips packets that you consume as well. Right. And RE digestion, all of you know, by the use of restriction enzymes. By the use of restriction enzymes, RE enzymes. However, the major drawback of these 2 methods is you cannot control the size. You cannot control the size. Now, why do you need to control the size? So you need to understand what is fragment size. What is fragment size? So now you'll have to understand about fragment size. You will have to select an appropriate fragment size for the desired read length. Now, all of us know from the DNA or RNA, you have generated fragments of different sizes by using the 2 methods that I have described. One is nebulization by using nitrogen gas. 2 is RE enzymes. These methods would generate fragments of different lengths. All these fragments will be of different lengths. From these fragments of different lengths, you need to select your desired lengths of fragments. Why I say lengths of fragments? Because here there is only a possibility of selecting a range of lengths. So you take all these different sized fragments, and then select your fragment of interest. So first you need to understand how do you estimate your desired length of fragment to be selected for generating your specific read length. First, let us talk about if you want to generate 2 into 75 base pair fragment. 2 into 75 base pair reads, not fragments, sorry. You want to generate 2 into 75 base pair reads. So ideally, this looks like this. You have a fragment in the middle, right? So you're going to have an A1 adapter here and an A2 adapter here. This is A1 and this is A2 here. So you're generating 75 base pairs here and you're generating 75 base pairs here. Ideally, if you don't want an overlap between these two, the minimum fragment size that has to be there should be around 75 plus 75. If you don't want an overlap, is around 150 base pair. 150 base pair. So people generally take paired end reads not to have an overlap between them. So they take an excess of at least 30 to 40 base pair in excess of the desired fragment length. So what is the fragment length that you need? The minimum fragment length is 150. You can add around 30 base pairs or 40 base pairs. So the fragment that needs to be selected, or the fragments that need to be selected, should be around 180 to 190 base pair fragments. Now, what do? You know that to generate 2 into 75 base pair fragments or 2 into 75 base pair read lengths or reads, you need to have a fragment size of 180 to 190. That is very clear. If you don't want to have an overlap, you can as well. Some of the cases refer to saying that instead of 180 to 190, even 150 is enough to generate 75 base pairs from both the sides so that you don't have an overlap between the reads. Ideally, it is 13 plus or 40 plus that may be needed. But nowadays, chemistries prefer exactly around 75 plus 75, that is equal to 150. But you should ideally know that the fragment length, if you don't want an overlap for 2 into 75, should have a minimum base pair size of 150. That is the take-home message. Now, how do you select this? How do you select this? Now you run a gel. You take a gel. Run a marker here. Have a marker here. Let us say this is 100, 200, 300, 400, 500, 600, and likewise. And then in the next well, here, you run your DNA sample. Run your DNA sample. This DNA sample that is run will run and you'll have a smear because you have fragments of all sizes. Now, what is the size that you need to select? If this is 100 and this is 200, right? And since the size of the fragment is 180 to 190, you can just go here and cut this particular fragment or cut this gel here from, let us start from 150, since we have taken 150 to be the ideal size. And you can cut this 150 to 200, take this out, isolate the DNA from this, and use these fragments for your adapter ligation. These fragments that you have got are now used for your adapter ligation. Got it? So that's how you normally do. What do? You take the digested DNA or sheared DNA, sheared in the sense of nebulization. Nebulization is nothing but high pressure use of gas N2 and digestion in the case of RE enzyme usage. You digest these and get these fragments, run on a gel, select this particular fragment, and use these fragments further for your adapter ligation. Now, the final method, since you don't have control over your fragment size, control on fragment size in the first two methods of fragmentation, that is nebulization and RE enzyme digestion, you don't have a control. You'll have fragments of all the sizes. Finally, you'll have to run a gel to select your fragment of interest. We'll do one more small example. Let us say you want to generate 2 into 150 base pair reads. 150 base pair reads means the fragment should be how much? 150 from this side and 150 from this side. That is a total of around 150 here and 150 here, a total of around 300 base pairs. A minimum of around 300 base pairs should be around this. What did I tell you? A 30 to 40 in excess would be better. So you can select 330 to 340 base pair fragments. And if you go back here and refer to that, since this is 300 base pairs, since this is 300 base pairs and this is 400 base pairs, you can go ahead and select a fragment somewhere between here so that you get your desired fragments of interest. That is running from 300, which is nowadays considered to be ideal, just enough to cover 150 from both the sides. And this was earlier taken in vogue that it should be around 30 to 40 base pairs in excess to the minimum requirement. So I take around this piece and then isolate these fragments, and then use these fragments for adapter ligation. That is what you use. This is one more example to identify or to actually calculate what is the fragment size that is needed for your adapter ligation. Since you cannot control the fragment size by these two methods, the current method that is in vogue is the third one, that is nothing but the bead-based method. Bead-based method. Now,